Welcome to the Fossils Lab. This week, our activities, content, and assignments will help you be able to do the following. Explore what a fossil is and how fossils form. Identify some of the common fossils found in Ohio. Analyze how fossils are used to study the history of Earth. Understand how fossils are formed and their importance in correlating rock formations. Recognize the scientific classification system for living organisms, and to identify fossils based on their characteristics. First, we'll start with some background information to provide you context for this week's lab. A paleontologist is an earth scientist that studies past life on Earth. The information that paleontologists have gathered about past life is called the fossil record. The fossil record helps us to understand past environments and evolution. Paleontologists use information from geology, biology, chemistry, engineering, and physics to reconstruct how an ancient organism lived. They study where it lived, like freshwater, saltwater, or on land, what the climate was like, how it moved, like crawling, walking upright, or burrowing, or whether it lived in herds and/or migrated. By studying the changes in groups of fossils over geologic time, we also obtain information on the evolution of those groups. One way we can do this is to look at the population of different organisms, or fauna, that lived at a particular time, in a particular area, or in a particular environment. Then, by studying how the organisms that make up the fauna change over time, we can learn a great deal about the evolution of life on Earth. A fossil is any evidence of pre-existing life. Fossils form in sedimentary rock. Typically, hard parts, like bones, shells, teeth, seeds, or woody stems, are the only parts of an organism that are preserved as fossils. This image of a fossilized Archaeopteryx is one of the world's most famous fossil specimens. Archaeopteryx was a small, bird-like dinosaur that lived in the late Jurassic period in what is now Europe. This specimen is from the Solnhofen limestone in Germany. There are body fossils, which are the remains of an organism, and trace fossils, which are the remains of the activity of an organism. Typically, body fossils provide evidence of the morphology of ancient organisms, or their shape. And trace fossils tell us something about the behavior of ancient organisms. Trace fossils are like the tire tracks at a crime scene. They can be footprints, tracks, trails, burrows, borings, or bite marks, or even fossilized excrement, like we see here as coprolites, which is the scientific term for fossilized poop. Most body fossils only preserve the hard parts of organisms such as bones, teeth, or shells. It is extremely rare for the soft tissues like skin, muscle, leaves, or flowers of an organism to be preserved. Taphonomy is a subdiscipline of paleontology that studies what happens to an organism from death to fossilization. Taphonomy has important scientific applications. The first is the importance of taphonomy to the fossil record. The fossil record is the library of fossilized species that scientists use to better understand what the world was like in ancient eons. However, there is a phenomenon known as the fossil bias that states that a fossil is more likely to be found if it is large, intact, and has hard parts that can be easily preserved. Between 98 and 99% of fossils found have a hard part in the body of the organism. This bias means that the fossil record alone cannot account for what life was like in ancient times. Forensic taphonomy and taphonomy in archaeology are also important facets of the field of taphonomy. Fossilization is a rare process. Most organisms, even those with hard parts, leave no record behind in the rock. Almost all fossils are found in sedimentary rock. Igneous and metamorphic processes normally destroy any trace of fossils. The process is so rare, in fact, that only 0.1% of all species that have ever lived have become fossils. Additionally, because soft tissues typically are not preserved in the fossil record, most fossils are incomplete. So what conditions are commonly required to form a fossil? The first is having hard parts, such as shells, teeth, bones, plates, or wood. The second is rapid sedimentation. This occurs most commonly in or near bodies of water like oceans, lakes, rivers, or wetlands. Low oxygen environments help to slow or prevent decay and decomposition, as most microorganisms that cause tissue decay, such as bacteria, require oxygen to do so. As mentioned above, normally only the hard parts of organisms are preserved, even during rapid sedimentation. To preserve soft tissue, not only is rapid burial required, but the organisms must be buried in a low oxygen environment to prevent decay of the soft tissues. Examples of this type of preservation are very rare. Worldwide, there are relatively few examples of such exceptional fossilization that preserve almost intact organisms, showing features such as feathers, skin, and even stomach contents. Sites at which these extraordinary fossils are found are known as Lagerstätten, the singular form Lagerstätte, which is German for place of storage. A spectacular example is the Solnhofen limestone in Germany, where the Archaeopteryx that we saw earlier was from. For our purposes, we are going to focus on three types of preservation: organic material, carbonization, and molds or impressions. Carbonized fossil remains, also known as carbonizations, may result when organisms are rapidly buried, especially in low oxygen conditions. Carbonized remains are thin, approximately two-dimensional films of carbon preserved on a flat surface of rock. They are most often black in color, reflecting the fact that they are composed mostly of carbon, as is coal, which is also black in color. Most fossils that exhibit soft part preservation are carbonizations. Examples include many plant fossils, also known as compressions, insect fossils, and the famous fossils of the Burgess Shale. Molds are impressions of an organism left behind in sediment. These can be internal or external. External molds are the simplest to think about, as they're simply impressions. Imagine pushing a modern shell into Play-Doh and then removing it. The impression left behind is an external mold. Internal molds are a little bit trickier to think about. Imagine a snail shell that has been buried in soft sediment. After the soft tissues decay, sediment fills the inside of the spiral shell. Eventually, this sediment hardens and the original shell dissolves away. All that may be left behind is the lithified coil of sediment that filled the inside of the snail's shell. This tells us what the inside of the snail's shell looked like, but it doesn't tell us much about the external morphology of the shell. The final type of preservation that we'll discuss in this lab is organic material. Organic material can be further classified as recrystallization, unaltered, or replaced, but for this lab, we'll just stick to the organic material classification. These types of fossils will look like a shell that will have visible thickness and potentially even a layer of mother of pearl. This is the original, unaltered material of an exoskeleton or the shell of a clam. Oftentimes, these fossils are not whole. The image to the right shows brachiopod fossils that had their original calcite shells replaced by pyrite. We will use three major concepts to classify fossils: whether the organism was solitary or colonial, the number of skeletal parts, and the organism's symmetry. You'll use your instructions, figures, and flowcharts to describe and classify the samples provided by your TA. This will allow you to classify the specimen by common name and phylum. Many fossils common in Ohio represent only one individual or solitary organism. If you see the repetition of major structures, the fossil may represent a colonial organism, which lives in a group of interdependent individuals. Examples of colonial organisms can include corals, bryozoans, and graptolites. A simpler way to think of solitary versus colonial structures: solitary organisms have one organism per body, as is shown in the horned coral on the left, and colonial organisms are one body with multiple organisms, as is seen in the tabulate coral on the right-hand side of the screen. Another major characteristic that is useful in identifying fossils is the number of skeletal parts. Remember, most fossils represent the preserved hard parts of an organism. Often, the entire organism is not preserved, and paleontologists must identify the fossil group from a fragment of an organism. Symmetry describes the arrangement of parts of an organism on either side of a plane, line, or a point. Most organisms are grouped into three basic symmetry types: bilateral, radial, or asymmetrical, as shown in the sketch. Organisms with bilateral symmetry can be divided by a single plane into two nearly identical halves that are mirror images of each other. Examples can include trilobites, brachiopods, and mammals. Organisms with radial symmetry can be divided into mirror images by numerous planes. These organisms tend to have distinct arms or partitions that radiate from a central point. In colonial organisms, each individual in the colony may have radial symmetry, though the colony may usually be asymmetrical. Examples of organisms with radial symmetry can include starfish and various corals. Asymmetrical organisms do not possess either radial or bilateral symmetry. Asymmetrical organisms may be coiled, spiral, or otherwise irregular in shape. Examples of asymmetrical organisms can include bryozoan colonies, sponges, and gastropods, also known as snails. When determining symmetry for colonial organisms, the symmetry of each individual member within the colony can be either bilateral or radial, and can be determined if distinct individuals are clearly visible without a microscope. The overall colony is likely asymmetrical. Fossil remains are classified according to the rules of biological nomenclature in a similar way to how present-day organisms are classified, which is shown in the diagram here. The main divisions of the classification system are listed in order from the largest grouping, domains, to the smallest grouping, species. As you move down the classification system from domains to species, you're dealing with organisms that are more and more closely related until you reach the level of species. In biology, a species is a group of organisms that can interbreed in a natural setting, produce fertile offspring, and are reproductively isolated from other organisms. Members of a species will share anatomical characteristics, or structures of the body and body parts, which are then used to identify them. Unfortunately, fossil species cannot be classified using the same criteria as biologists use with modern organisms, which is whether two individuals could have interbred and produced fertile offspring, because that's impossible to determine in the fossil record. As such, paleontologists use morphology, which is the form, structure, or the shape of a fossil, to classify specimens. In this lab, you will only be classifying the specimens to the phylum level of a major group, not to the genus or the species level. One of the major uses of fossils is to determine the relative ages of rocks. We discussed that in the geologic time lab. Rocks found at the bottom of an undisturbed sequence are older than the rocks found at the top of the sequence. Certain fossils are found in different sedimentary rock units. By identifying these fossils, individual rock layers can be identified over large areas. Slowly, the age relationships of rocks from one area can be extended to nearby areas. We refer to a fossil that is useful for dating and correlating the strata in which it is found as an index fossil. To be used as an index fossil, the fossil must have a wide geographical range, but a short, distinct time span of existence. For example, by using rock characteristics in the fossils contained in the Redwall limestone that is exposed in the Grand Canyon, the rock unit can be traced, or correlated, throughout the western United States. Over 700 miles away, fossils indicate that the Madison limestone in southwestern Montana is the same age and was deposited in a similar environment to the Redwall limestone in Arizona. In this example image here, the coiled shell is the good index fossil, because it only shows up in one rock formation or layer, not multiple, and it's present in each location or each outcrop. Next, we'll discuss information that you'll need to complete this lab. There are no safety concerns specific to this lab. There are no helpful hints specific to this lab. Fossil specimens will be provided by your TA. Additionally, worksheets and procedures will be provided by your TA, but will also be available on the instruction page in this week's Carmen module. The procedure and flowcharts will be provided by your TA, but these will also be available on the instruction page in this week's Carmen module, so they can be downloaded as digital supplies. Please let your TA know if you have any questions.